So we're next going to talk about something called cell signaling. And when we have a multicellular organism and we have gene regulation going on, cells need to communicate to each other, and cells can also affect the gene regulation of other cells by this means of cell signaling, okay? So let me just say, within a multicellular organism, information must be communicated between cells. Okay? Cells can produce and secrete chemicals. Some of these are going to be called hormones. That affect gene regulation in other cells. Okay, so this happens a lot. The whole menstrual cycle is based on this idea of cell signaling, where we have parts of the brain that are sending out signals to the ovaries. They're telling the ovaries to start to develop an egg. While that's happening, the uterus is being readied for implantation. There's lots of different chemical messengers involved, hormones. And so this is how, you're having regulation of different genes. So you're saying, like, you need to turn on these genes for this to happen in this cell, okay? And so there's this cell signaling going on. So the way that this can happen is that you can have just a single molecule can act by binding to a receptor protein and initiating a signal transduction pathway. Okay, and so what this basically is referring to is a message can come in, and then that can be translated and moved to the DNA and affect what's happening to the DNA. So I drew this out before. Okay, so this could be our signal molecule right here. So it could be a hormone, it could be something else. We then, on the surface of the cell, can have a receptor. So the signal molecule will come into the receptor, it will bind to the receptor, and what that will then do is cause this chain of events to happen. We call this chain of events the signal transduction. So we're moving a signal from one location to the other. Ultimately, what's happening is we are then going to produce some type of molecule that could go bind the actual DNA and then causes something to be transcribed. Okay, and so in this case, I'm showing that this kind of boomerang-shaped structure can then go in and affect the DNA and potentially cause transcription to take place. Okay, so we have reception of a signal, we have signal transduction, and we do that through these relay molecules. So these are going to be proteins. And then we can have response, could be gene expression, and that's happening in the nucleus. Okay, and this is happening all the time. So this is going to be a really important process. Okay, now moving on from there, we're going to start talking about kind of how cells become specialized. And if you think about the human body, we have many different types of cells. We have skin cells, we're going to have muscle cells, we have neurons, pancreatic cells, liver cells, all these different types of cells. And the way that they become specialized, because remember, they're all going to start off from that single zygote, is through a process called cell differentiation. Okay, and so think of cell differentiation as how do cells become different? Okay, so a multicellular organism develops from a single cell, remember that's a zygote, into a collection of many different cell types organized into tissues and organs. Okay, so this is cell specialization. So cells specialize. Okay, and what they're actually doing is that they're having what we call, they have different patterns of gene expression. Okay, and what this means is that they will have different genes turned on and others turned off. Okay, so the pattern of gene expression will guide the behavior. Okay, so a cardiac cell, one of its main jobs is to be able to contract and help the heart pump the blood to the other parts of the body. The pattern of gene expression in a cardiac cell is going to be different from that of a skeletal muscle cell, or a skin cell, or a neuron, or an eye cell. All those are going to have different patterns of gene expression. Okay, so cell differentiation happens through these different patterns of gene expression. Okay, so the cell differentiation is kind of cells taking on their final identities. So we start off as this one type of cell and can ultimately turn into all the different types of cells of our body. Okay, now what this brings up is that this idea of this genetic potential of cells and how the initial cells in our bodies can potentially turn into any cell in our body. Okay, so we're going to call this the genetic potential of cells. Okay, and so what this is going to have implications for is cloning and kind of regeneration. This idea that if you have cells that can turn into anything, so if you have a cell that could turn into a neuron or a muscle cell or a bone cell, it has this endless possibility of being able to regenerate different parts of our bodies. Okay, there are multiple problems, and we'll talk about some of them. But this idea of being able to tap into this undifferentiated cell that can turn into anything is a really exciting bit of science, and potentially is, how we're going to be able to cure different diseases in the future. Okay, so gene regulation will affect both cloning, and we are also going to talk about cancer, because this will be part of this. Okay, so all body cells contain all the same genes, which we've said multiple times, even if some are not expressing all of them. One thing that we would love to be able to do is to take cells that are able to regenerate very easily, like skin cells, and then turn it into maybe a neuron or nerve cell. So certain types of cells do not regenerate, and if they're destroyed, basically there's nothing you can do. And so if you have kind of a spinal cord injury or have part of your brain that has been affected by a stroke, those cells won't regenerate. And so if we can take cells that could regenerate them and program them so that they can turn into a different type of cell, like a new neuron, that would be kind of this holy grail of medicine. Okay, so we could grow you a new kidney from your own cells if we knew how to do it. Okay, and at this point we can't do those types of things, but this is where the science is going. Now, we know that this is a potential because we know that certain types of organisms like plants can actually, you can take a single little cell removed from a plant, you can grow it in a culture, this growth medium, and basically any part of that plant can then, you can turn it into a brand new full-fledged plant. Okay, so this is the idea of cloning. Okay, so a single differentiated plant cell, so it's already been turned into, let's say, a stem cell, or I shouldn't say stem cell because that's going to be petals, let's say leaf, a plant leaf cell, can undergo cell division and basically give rise to the full adult plant. Okay, so you can take cells from an orchid and grow a new orchid if you really want that specific orchid type. Now, this doesn't happen in humans. You can't take off a piece of my finger and make a new me. It's more complex than that. But there are certain animals that can have some types of regeneration. So, actually, I'll do it on this page. So, for example, right here, this is the progression of a salamander arm that has been cut off. So this is the normal arm, and then it was chopped off. And then they gradually, over time, took pictures and watched it until it regenerated this foot. Okay, we know that in humans, we can't do this. If people lose their foot or their arm, they don't get another one back. They would have to have some type of prosthesis. But this idea of being able to regrow lost body parts is something that's very intriguing to humans, okay? This happens in lots of invertebrates, so less complex organisms than us, and there are some simple animals. So we have, like, lizards can grow a new tail. Okay, so these are going to be capable of regeneration. Okay, so within these cells that are present on this limb, they have the potential to produce new limb bones, new skin, new tendons, new muscles, okay? Now, what has been done with cloning is that we've done some things called reproductive cloning. And this is first done in animals. Okay, we have not cloned a human, but the way that this takes place for do reproductive cloning is that we do something called nuclear transplantation. Okay, so this involves replacing the nucleus of an egg cell. Of an egg cell or zygote with a nucleus removed from an adult body cell. So this was done in sheep. You maybe have heard of Dolly, although at this point this is getting old enough that some of you were probably born after Dolly took place. I think Dolly was in 1996. I'm not positive about that date. But this is how this worked. They had a cell, an egg cell. They removed the nucleus, okay? So they could take the cell from any sheep. They then got a donor cell from the specific animal that they wanted to clone. They put the nucleus into this egg cell. They made sure that it was growing. Then they inserted, one way to do this is they insert it into an actual sheep and let it develop. So it's kind of like a surrogacy where, you know, it's not her egg, but it's her oven, you know how kind of that works. And when she gives birth, it will be a clone of the donor, kind of where the nucleus came from. So it should have all the same identical DNA as the donor, okay? Now, this does happen, and we use this for specific things, but there is another potential where you can actually take some cells from this growth, and then culture them outside of the body and try to make different types of cells. So, if you want to make cardiac cells, you could, if it's under the right conditions, these cells could turn into cardiac cells. Okay. Now, why do we do this reproductive cloning specifically of animals? There's a couple different reasons. First of all, it helps us understand this whole process. And, if we want to get to the point where we can start making new organs for individuals from their own cells, we have to know how to do this type of cloning. Cloning used. Okay. So first, in agriculture, this is used. So if there are animals with specific traits, set of desirable traits that they really want to have, then they can do this. I've heard of people doing this with dogs. This is a good way to make experimental control animals. So if you need mice that have a very specific genetic makeup, you want them all to be the same, you could do this type of thing. This is also for pharmaceutical type of things. Okay, so to be able to ultimately try to make cells or make an organ that you could then transplant to someone. And they also have done this for cloning reproductive cloning used to restock populations of endangered species. Okay, so if we have species that are really endangered, this is one way to kind of help their population. Now, the big thing is that this always leads to the idea of why don't we clone humans? Should we be able to clone humans? First of all, the cloning of mammals is extremely difficult. It's extremely inefficient. It doesn't work most of the time. I think it has like a 10% success rate. It's very expensive, and it's just not something that people are going to be able to do, very often in labs, and the money's not there for it. There is, however, money on the therapeutic cloning sides and this idea of creating things called stem cells. Okay, so we have to talk about what stem cells are. Okay, so let me just put, this is therapeutic. Did I spell it right? Therapeutic cloning and stem cells. Okay, so one of the big goals is to be able to produce something called embryonic stem cells. Okay, and so I want to take you back to the very beginning of this lecture where I talked about how you have a zygote that ultimately will divide and then continue to divide, so on and so forth, to ultimately until you get to, the full-size person. Okay? What happens is at the very earliest stages of the embryonic development, these cells have ultimate potential to turn into every single type of cell. Okay, so these cells could turn into every single cell that you would need in your body once you're an adult. What happens is that as we grow and as the cells become differentiated, they lose that ability to turn into anything. And so cells become fated for specific types of functions. And so at some point you're going to have cells that turn into, they'll be fated as blood cells or nerve cells or muscle cells, and they can't be turned into other types of cells after that point. Okay, so embryonic stem cells are what we call undifferentiated. Okay, they're undifferentiated and they can divide indefinitely and under the right conditions can develop into a wide variety of specialized cells. Okay? And if scientists can discover the right conditions, you potentially could repair, okay? So you could grow cells for the repair of injured or diseased organs, okay? And so this idea of being able to actually take your own cells and making a new organ for yourself to replace something that's been injured or diseased would be, you know, it would change everything basically. At this time, if you have an injured organ and it's a type that can have a transplant, you have to make sure that the person has you're not going to have some type of immune reaction to it, or your body is going to reject it because it's not yours and your body rejects things that aren't itself. And so if you're able to have your own stem cells and create new organs, it's your own body and your body's not going to reject it. So this is really kind of where medicine is going and trying to understand how we could make new organs. Now, the problem is that embryonic stem cells are extremely controversial because people think that some people think embryos are considered living things and that you can't manipulate them. And it's, there is lots of different kind of religious and other problems going on with stem cells. And so there often have a lot of controversy surrounding them. There are ways to save stem cells, and stem cells don't need to come just from, embryos. They can come from the umbilical cord and things like that. But it's expensive to keep umbilical cord stem cells because you'd have to freeze them and keep them in some type of very regulated situation. But the other idea is if you could actually take adult stem cells, and adult stem cells people don't have as much issue with those. So we have adult stem cells. There are certain cells that are present in the adult human body that are somewhat undifferentiated, but they are not embryonic stem cells, so they're not quite as good. And so they can then give rise to some types of cells. So I can show you what this looks like. So if you have embryonic stem cells, so that you would take this from an early embryo, you can culture those embryonic stem cells and basically then produce all different types of cells. Okay, so you can make blood cells, nerve cells, heart muscle, they can turn into anything. However, again, these are controversial. If you can take adult stem cells, so you could take maybe bone marrow cells from an adult, they have the ability to produce blood cells. However, they could only produce blood cells. You can't take adult stem cells at this time and turn them into muscle cells or nerve cells, okay? And so they are already specialized in that they will produce blood cells, which is important, maybe that would be beneficial, but they only have so much potential. They cannot, again, be turned into heart cells, heart muscle cells, or nerve cells, okay? So scientists are still trying to figure out if you can take an adult stem cell and turn it back into an embryonic stem cell, but it's extremely complicated and it just doesn't seem like you can reverse all the steps that happen during specialization. Okay, So kind of switching gears a little bit from this idea of stem cells and cloning, and we're going to talk a little bit about cancer and the genes that cause cancer. Okay, and so when cancer occurs, what often is happening is that the cell will escape the control mechanisms that regulate growth and division. Okay, so the cell cycle is tightly regulated, and we want to make sure that cells aren't continuously going through mitosis and producing a lot of them. And so unfortunately, when cancer starts to happen, the control mechanisms get kind of out of whack, and the cells are no longer following the rules, basically. Okay, the way that the cells escape this, so we have cell escape involves changes in gene expression. Okay, now one of the earliest discoveries of how this happens is when they discovered that a cancer was actually caused by a virus. Okay, so one of the earliest clues to the role of genes in cancer was the discovery and this happened in 1911 of a virus that causes cancer in chickens. Okay, and so what happens is that the virus, or viruses that cause cancer, and there's a lot of them that do, can be permanent residents in host cells, not hot cells, host cells, by inserting their nucleic acid, basically inserting their genetic molecules. So sometimes they're DNA, sometimes they're RNA, into the DNA of the host. Okay, so viruses will actually insert their own genes into the DNA and will be they can be permanent in there, and they often can, if they're in the right spot, can cause cancer. Okay, now genes that cause cancer have a very specific name. Okay, so a gene that causes cancer is called an oncogene. Okay, so an oncogene is a gene that causes cancer. And before that, so a normal gene with the potential to become an oncogene is called a proto-oncogene. Okay. Many of the proto-oncogenes are going to be involved in the cell cycle. Okay, so many proto-oncogenes code for growth factors. Okay, and these are growth factors are proteins that stimulate cell division or affect the cell cycle. Okay, so when they start to malfunction, cancer can start happening. Okay, and for a proto-oncogene to become an oncogene, a mutation has to occur. Okay, so here's a proto-oncogene, and this, for example, would code for protein that stimulates cell division. And this is kind of, if we have to have a mutation within a gene, the oncogene can turn into an oncogene and then have this hyperactive growth stimulating protein. If multiple copies of the gene are copied, so like there should be one, but now there's 3 of them, you can have normal growth stimulating protein in excess. Okay, so not necessarily turning into an oncogene. And then this mutation within a control region of the DNA, you can have a mutated promoter, and that could have normal growth stimulating protein in excess. So all of these can start to lead to kind of too much cell division, which could then lead to cancer. Okay, now the mutations that can take place changes in the genes whose products inhibit cell division are also involved. And so we can have, there are these genes called tumor suppressor. Okay, and so these normally, okay, they're proteins. They encode, normally help prevent uncontrolled cell growth. Okay, and if there's a mutation in it, that keeps a growth inhibiting protein from being made or from functioning, may contribute to the development of cancer. Okay, so again, what's happening in this and why this is in the gene regulation lecture is that you have these genes that are involved in regulating specific other gene cells and other genes. And if that regulation goes haywire and stops working correctly, you can start to have issues. So here's a tumor suppressor gene. So it's normal when it doesn't have a mutation, it's a normal growth inhibiting protein. It makes sure the cell division is under control. If there is a mutation in the tumor suppressor gene, that means it's non-functional, which means it's not being able to do its job and inhibit mitosis and cell division. Okay, and so that can then lead to cancer. Now, what happens is that there are usually kind of once you start to have one mutation, other mutations are more likely to happen. So for example, in this case, an oncogene is activated, which then increases cell division. Then if there's a mutation in the tumor suppressor gene, it becomes inactivated, then that cell division can continue to grow and become a benign tumor. If there then is a second tumor suppressor gene inactivated, that can then lead to the growth of a malignant tumor. So what happens is you have a normal cell, but then if you start to have mutations, the accumulation of mutations then can cause cancer, and then ultimately could lead to malignant cells, which then can move from other parts of your body and cause major damage. Okay, and so all of this is going to be involved in this gene regulation and helping to kind of regulate when things are going to be made and when things aren't. Okay, and so we will move on to DNA technology next time.